cloud, it started raining.
We are conducting for serving the growers across the Commonwealth of Virginia and beyond. So I titled my presentation today as expanding the toolbox. The idea is that we would like to provide different control tactics for growers to be successful at their businesses. So I'll start acknowledging an army of people, army of institutions, because what you're going to see here, it's not my work entirely, but it's the collaborative work of not only the growers, but also the funding agencies that I will be able to, and the names that you see there, that I've been closely collaborating with all these different projects that I will give you this executive summary in this presentation. That's my team. I want to first and foremost recognize them, because without this amazing team, I am not be standing here. I will not be running the program that I run. And here's that was the National Harbor and the ESA, and that was actually in Western New York, Niagara County, where we were finishing putting a mating disruption trial that you guys are going to hear about today. Always smiling. That's key. So, as Cesar mentioned, I am running in a specific program where I would consider working on topics that are applied ecology. So I use ornamentals and turf grass as the study systems to understand the ecology, and when understanding the ecology and biology, we would like to propose the science-based solutions to the growers that are affected by pests, and the idea is for them to be successful and keep those profits. So within the areas and the research areas that my lab works on, and the team that I have, we are interested in biology and ecology of insects. We are also interested in control tactics, and especially on the biological control. And we also are really interested in alternatives to broad spectrum insecticides, and keeping in mind that some of the registration for specific chemicals are actually going away from the use for the growers. So the idea is that can we propose an alternative for them to use when chemical control is warranted. This is a diagram that resonates a lot to me. Surendra Dara, who is now in Oregon State, we both were in California when Surendra put this together, and actually it's also trying to summarize how complicated IPM could be. So I'd like to show them to the growers. I'd like to show them today. I have the podium today, so the idea is that I can say a couple of things, and this is what summarizes what IPM in the sense that resonates to me. So IPM and the core, the heart of IPM, research and extension, are what it's actually make them the system be. Then you have the regular, what it's expected, the pest control. Then you got knowledge and resources, you got planning and organization. I keep telling my growers and the superintendents that runs the golf courses is, even if you're texting to your colleague, hey, what product do you use? What pests are you seeing? What are the densities you're seeing? You are practicing IPM. Because the idea is that you're moving information without knowing that that's going to help to be successful, to be on time and understanding the system. Also, I like to point out the reality of IPM. The reality of IPM has a purpose to at the ring of the business aspect. And it comes close to that entire organization. Ultimately, growers need to make profit. If they don't get payroll, they will be laid off. So why are you going to implement a system, an IPM strategy, if you are not successful? And then obviously, because more people was like, well, Alejandro, but environment is also important. Yes, it is. That's why we have the sustainability aspect. But again, some people might disagree with this view because ultimately when you read the 67 definitions of IPM, a 3-word concept that has more than 60 definitions, you can see how those specific keywords are starting moving and being put in together, right? But this is great. So I use this some sort of a guided map on what I would like to conduct research on. So I start with, well, let's study what is actually causing the problems, right? Let's know our enemy. So there's a bunch of different specific topics that you're going to hear today where we try to understand the biology and ecology of pests. This is a beautiful shot, one of our collaborators. Right there, you have 1, 2, 3, 4, 5, 6, 7 different plant taxa that you can see. So nurseries and ornamentals are intrinsically diverse. So I was already overwhelmed when I started my position. What insect I'm gonna study? On average, a nursery grower will manage 150 plant taxa, and every single plant taxa is gonna have an insect. I'm never gonna finish trying to study an insect per plant taxa. This is just, and then the other part is, I learned from the books, and when I was a student, that if you are diverse, you shouldn't have pests. What is happening here? The insects are not reading the textbooks that we write about them. They're pretty smart. Insects are pretty smart. But they know, they pretend that they don't know. But look at the nursery and ornamentals. We got diverse system with hundreds of plant taxa being used at the same time. Those are annual multi-year crops. And also it is one pest of interest for each one. Well, thankfully, I went to the source. I went to the growers and I asked them. I put it in a room and say, hey, what are your top priorities? And they were so clear. Alejandro, we need really help with the red-headed flea beetle, which was at the east part of the state. And in the center and across the state, they have problems with a specific scale and an ambrosia beetle. And now there is a threat for the entire industry in the country about the newly invasive bostrom. All right, that keeps me sane. I am not going to be losing sleep about thinking 100 insects that I'm going to study, and now I can do 4. The first one, the red-headed flea beetle. The red-headed flea beetle is the key pest in the southeast right now for ornamentals. This beautiful beetle. To me, it's beautiful. My growers, they hate it. Every time I'd refer as a beautiful beetle, they just like roll the eyes automatically. Beautiful beetle. Cisterna frontalis, native in the Americas, first reported back in the 1800s. This is how you put one beetle in a petri dish with a hydrangea leaf. This is how you're gonna see it. It's entirely different. These guys will lay the eggs on the potting. So you have containers, right? So if I'll show this to you. You see there's all these containers, right? And every single container you have the potting media. That potting media is used for the red-headed flea beetle to put the eggs and actually complete the part of the immatures. So the immature is going to be feeding on the roots. There is no direct damage, well, that we can quantify as damage. There is injury from those immatures feeding on those roots, but we don't see the plant stress enough. Because if I wanna put something in perspective, these are systems that are getting high numbers of inputs, water and nutrients. So you can compensate when you start seeing those yellowing stress. All right, let's put in some nitrogen, right? So we can mask this, but that defoliation cannot be masked. So the growers are really battling this because they were spraying. When I got there, they were spraying 3 times a week. Now they're doing 2 times a week, and the goal will be to spray once a week. But hopefully, with science-based solutions, they will head into that direction. So the first thing they want to understand is when they are active, when that happens. So basically, the phenology. So this is Julie Brindley. who helped me with the lab, you can see her going through a hydrangea block using a leaf blower on the vacuum part, and we just collect the beetles for 5 plants, and then we assess the defoliation for those plants. So we got 4 nurseries across the state. We were doing that technique for the larvae. We actually, and then half of the group is gonna take the pots, take the containers out, look through the potting media, and then count the larvae as well. So it was highly intense. We really need to understand what's happening in the system. We were blind. All we know is the beetles will show up, defoliate everything, and you have to spray. Nothing else, right? So the idea is we wanted it to put a start line, say, all right, let's understand when these beetles are in the system. So this is what we learned. We run this actually 4 years, but we were able to publish this. And we learned that there is 3 generations in our systems. And I like to show this to the growers because they are, okay, Alejandro, there's 3 generations. The first generation is going to start May 1st. We're going to spray the entire farm May 1st. I was like, take a look at the data. The data tells you, sure, you can say May, but then it will move, the peaks can move. And why is that? Because there's weather parameters, things that you're growing. So it's a dynamic system, and the data tells you that one of the things is, please, scout. Alejandro, I don't have those vacuums. Well, it's plain and simple. How about this? How about if we show you when the beetles are active? So this is Ellie Lane's, my first master's student. She did phenomenal work where she was able to track the activity for the beetles. At 1 in the morning, 3 in the morning, 5, you can see the x-axis. This is really kind of like, I remember when Ellie and I we were talking about it, she's at the beginning was like, okay. But then the growers like, oh, so we're going to know when these beetles are actually moving. And we were able to document that between 11 in the morning and 3 p.m., you're going to see them active. So now the suggestion that we give the growers is before you go to your lunch break or right after your lunch break, go to those specific corners of your farm that you know or you might know you have a hotspot. Walk that block and see if they're fleeing and they're moving because they're going to be active. No matter what you do, they're consistently active there. So to avoid underestimation of what the problem could be, you can target those. So don't tell me, please, because everybody's busy. That's the new norm. Everybody is busy. But within your busy schedule, you're not going to spend so much time. So what happens if you go at 7 or 9? When you do have the time, because, you send all the labor out and you are like, okay, I have time now because everything started at 5:30 in the morning. But at 7, they're not going to be there. So you're going to really need to dig and scout. But what happens if you start looking when they are actually active and not sedentary? You will be able to spot those hot spots and then treat them accordingly. So that's the idea about applied research that we were trying to promote with the growers and give them some directions on how to manage this. The next one, my lab and myself, I'm really interested in landscape ecology. And one of the questions that we were always asked is like, how far these insects can fly? And I'm going to show you a little bit of we use fly meal, but also the mark and recapture technique. So that's a red-headed flea beetle being overly dusted. So we went crazy. So just for the purpose of showing what it's supposed to with this fluorescent powder. So we collected over 1,000 beetles from a commercial block. We marked them with the powder and we released them. We were not bringing more beetles into the system. These beetles already existed. The grower was standing there and said, Alejandro, you just collected 1,000 beetles from my block and you're going to put them back. It's like, well, they were there. You're going to spray it in 2 days. They're going to die before you kill them. I can study them. It's like, okay. So we have a center of the block and then we mark 5 feet and, in transects, 5 feet, 25 feet, 50 feet, and 100 feet. We have those GPS marked. And then we went 24 hours, 48 hours after we marked, and we collect those beetles. So at the end, from the 1,000 beetles that we capture and marked and released, we collected 106 total. That was pretty good. I was excited. I was expecting 3%, and I would have been pleased. And if you read the literature about this, 3% is like, whoa, 9% were, but well, I'll show you what. So when we did the analysis, red-headed flea beetle won't move that much because every single where it goes is going to have a nice food source. Why are you going to invest energy in moving since you are surrounded by all these volatiles, physical cues, and everything that you have? Just like, this is great food. I don't have to go, right? So they were close to the release center. There's east, west, north, south. And then the y-axis, where it's the positive and negative, is the amount of beetles we capture. Like there were higher numbers, cluster within that 5 to 50, but they were able to go all the way to 100 feet. So this data, we took at it. So what I do is something that I name it now, the IPM schools. So I go to the growers' farms at the end of each season, and I present the data they help us to collaborate with. And this actually opened the discussion with the growers while we're sitting with the entire team and we're presenting this information to them. It's like, all right, Alejandro, we are ready to start creating physical separation. What happened? The common practice in nurseries and ornamentals is that you have a newly potted hydrangea or a plant next to an old hydrangea. So guess what? The beetles just go back and forth, back and forth, back and forth. You keep spraying, you keep spraying. So now, what happens if you put your new hydrangeas 100 feet away from the old hydrangeas? You're reducing the risks from exposure to recycling your same population. And I have already one grower that it's been doing this consistently because we did the flight mill and we were able to know at least in theory how far they can fly, which is over 500 feet. It was really surprising. So this grower said, all right, Alejandro, I'm going to do this physical separation. He's the only one that's spraying only once a week. Compared to the other growers that keep the traditional ones and they're doing twice a week. So now we're promoting this information, saying, hey, could you consider that 2 years ago, this was a pushback. In nurseries, every single inch square, it's a precious cargo. You don't have room to move things around. That's a luxury. But now there is an insect that is costing thousands of dollars, and not only in control, but also in the orders that you cannot ship. So I think now we're ready to start moving and implementing new things. That was the red-headed flea beetle, and we continue to understand that we have active projects about how irrigation, overhead irrigation, if there is any insecticide resistant that might be able to. So there are multiple projects right now that we're running. To be determined that we will learn more things about this beetle. The second pest, Japanese maple scale. Scales in general, I've been fascinated about scales. This one actually hit really hard at the central part of the state, where 3 big growers were suffering through this pest. This is a boxwood, thin, heavily defoliated with Japanese maple scale. So basically you start seeing canopy death in terms of foliage and branches and things like that. So this is just to give you an idea, that's an infield grown boxwood that it's 4 feet tall. It takes 5 years to grow that. And that single one, it's potentially the cost, you sell it around $250, $300. And if you think there will be hundreds of acres with this boxwoods in there. So do the math, and that's what it's happened, right? So this was a big problem over there. We expected to see the females as being the overwintering part. And unfortunately, the presence of this pest will deny the shipping of in a specific order. So you have to go through a certification. If the inspector sees GMS, Japanese maple scale, they say, well, no, we're stopping the shipment. So it will go from, all right, we're ready to stop the trucks. We need to spray And it's really detrimental. So, and also expected to have 2 generations, one in May and one in August. Just to give you a overall idea, here is a female that was flipped. Those are the eggs, so the round pinky structures there. The next one that's a little blurry, that's a crawler. So in this case, the scale have 2 life stages that are going to be moving. Then it's going to be set and still once it's established. So the first one that moves is the crawler, as the name implies, it crawls out of the female and then redistributed, established, and settles. And then the second one will be the males that have wings, and those that close with the wings and gonna follow and find females. Females are the ones that stay on the bark, and that's what they're really obvious. Then the crawler, once settles, creates a shield. It is a waxy shield. That's called the test. And then when you flip that, you're gonna see the immature growing, and then they will continue to grow until they get adults. And then you have this differentiation between the females and the slender males. They're going to be actually having the wings, and then it close with the hole of eclosion, and they're going to be flying to find the female. So see, it's an already complicated system for even trying to not only understand, but also manage it. Again, I'm going back to the same comment. The insects are pretty smart. Book says that they overwinter as immature females, where we start collecting data from the grower, one of the growers, mid-January, mid-February, mid-March, and the hypothesis was we're going to see a consistent density of immatures throughout the time of the winter. And then when we get close to the April time, everything's going to start going to adult. But guess what? That's not even happening. They are there, and they start developing through time. So why is this important? Because growers were, with that information, with that previous information, they were spraying, because it's immature that you expected, insect growth regulators. What happened when you spray insect growth regulators to these adults that are coming into the system? And then I got a phone call. Alejandro, I just sprayed IGR, insect growth regulator, nothing's happening. I was scratching my head like, whoa, but they were supposed to be immature females, right? I showed you the book. And they're like, yeah, this is what we're doing for multiple years, because that's what they say. It's an immature female that's coming. So I targeted the first thing that I wanted to do. Guess what? They're like, no, we're already developing. But if you think about it, I was corresponding before coming here with Carla Deso. She's a phenomenal entomologist as well. They're dealing with Japanese maple scale in Tennessee. We're talking about the canopy structure, how the temperatures are going to change, how they're so protected inside the main trunk that this could be actually affecting their actually growing degree days and promoting the idea that they can continue slowly and steady. They are developed. This is key. Now they do both. They do IGR with a complementary, which is something that is going to affect the adults as well, a systemic or something. And they, unfortunately, the guy I was working with, he doesn't have GMS problems anymore. My student barely can make collecting data because he started to implement these different things to understand when the crawlers were present. Then yeah, Molly, we made it, but yeah, there was Molly Wyatt who helped me with this project. But it was very, very interesting to learn that this is actually something very dynamic and changing. Well, Molly helped us to understand where exactly, because we use electrical tape as a way to scouting. The sticky side, you put it up, you banded the trees, and then all you see, those are crawlers. And then you target your applications one, you see the crawlers coming out. Now the question, there is always an applied question. Alejandro, you're suggesting to put a tape. Where should I put it? And I always say, wow, that's a heavy loaded question. Wow. If I tell you to put it in the top, if I tell you to put, and if you don't capture it, and then this one is nested with each other because, the ones that are coming from here, but that. So at the end, we were able to see consistently after several experiments is that the middle portion of the canopy is where you're gonna most likely gonna have the success to capture the immatures. Very interesting. That's one of the designs that Molly had in the field with all the lines and things to trying to keep us straight with what sections of the canopy we're gonna do, internal, north, south. It was very, very, very interesting. Now, throughout the state of Virginia, Commonwealth, I would say, I've been corrected multiple times that I don't use the proper term. The Commonwealth of Virginia is that ambrosial beetles. These guys came in the 30s, and now become, prevalent in the system. Here's the typical damage that you're going to see. There's toothpicks strutting out, and ambrosial beetles are actually not feeding on the plants. I like to tell my growers, and I would like to tell you, the ambrosial beetles use these trees as Airbnbs, fancy Airbnbs. They just be there. They use it as a protectant. They bring their own food. They're really good. They're really good because they bring their own food with the mecangium, and then there's fungus that they're gonna put it inside the galleries, and then the immatures are gonna feed. Females are the only ones that flies out of the galleries. The males stay there. This is a potted redbud that you can go to Home Depot and Lowe's and buy it. Would you buy that one? No. How much is a redbud as a 2 or 3 year? 150, right there. So you see now the problem, right? This is the straight influence in what the profits are. How do the ambrosial beetles find the trees? Well, they have an affinity to ethanol, and I don't blame them. I love ethanol. I don't know about you, but I do, and they do too. Ethanol is so powerful. Look at this one. In Louisiana, gasoline mixed with ethanol was in a cantineer. This is a peer-reviewed published paper. This is the photo from the paper. This is the rear ends of the ambrosial beetles burrowing into the plastic because has ethanol in it. You're enjoying yourself in your patio or in the bag, and you sipping wine, and all of a sudden you see one thing landing on your wine glass. Well, the ambrosial beetle thought that you are actually drinking a tree. Same for beer. These are real photos. And I'm pretty sure you'd be laughing because it might have happened to you, that you have this. I was like, oh, okay. Well, ethanol, it's a byproduct. Ethanol is a byproduct when you're stressed. There's anaerobic processes within the plant that results in ethanol, and ethanol gets emitted. So the more stressed you are, so because of the tree got cracked, or it was a really cold winter, so you got freeze burn, or there is a flooding event where there's a lot of water and you get stress, automatically you signal ethanol, and that ambrosial beetle cues on that and say, okay, so if you're stressed, you're not gonna fight me back. Basically, I'm gonna know the keypad, I will know the combination, I just come in, compared to these fancy things that I won't be able to if you're a very healthy tree, right? Ambrosial beetle has been studied for a while, not necessarily in Virginia. So that, again, we decided to understand what are the species of ambrosial beetles, when they are present in the system. So we have this set of traps. We started with lingering, that we move away from all these traps to a 2-liter soda bottle that costs you 50 cents. Because when we present it to the growers, it's like, I'm not gonna pay $20 for a trap. Okay, how about if you pay one, it's 50 cents. It's a 2 liters, and you can drink the soda, and you can cut it yourself. And now you have the ethanol lure. You put it in there, and you have your trap that tells you when the ambrosial beetles are flying, so you can time properly your applications. That is the overall idea. And then we also use 1-foot pieces of wood. In this case, it will be a branch. We core in the middle, and we put ethanol, and then that helps us to understand how many of those beetles that are flying are going to be able to get into a piece of wood. So those are 2 types of traps that we use, the wood bolts and also the soda bottles. From the soda bottles, just to give you an idea how intensive this is. We have identified up to 24,000 beetles, every single one. I helped. Believe me, I helped, but I haven't. You see why my team is so amazing? You're counting about it consistently. Doesn't matter. The most prevalent species in Virginia, it's one that covers over 70%. Then we have the rest that can change through 2 years because of, the landscape changes, so many other factors. And then these are the dates that we were putting the traps out, so that can also be in the number of locations that we use as well, they can change. I am proud to show you the most expensive graph in my entire career. This is 21 years of data for ambrosial beetles for Crassisculus, one of the species, in one location. So the credit goes to my predecessor, Pete Schultz, who started that, had that vision about it. So we are confident enough to say from April 10, so sometime in April, all the way to May, you are going to see a first peek of it. This actually is the roadmap for our trials. Every time you set up a trial for us, so we're getting ready, for example, right now, to start deploying traps based on this data. Because the traps that we deploy or the trials that we're going to set up, we're going to need the presence of the ambrosial beetles that are coming pretty soon. Now, there's a second peak, but that peak is not necessarily what we call economical. Now, with all that data combined, and that's the bolt on the right, the piece of wood at the stopper, we put the ethanol, it's 70% ethanol, and then we leave them for a week. The traps are also checked every week, so the trap actually has a collecting bottle. Right, and we have an entire system. We use coffee filters and the suction, and then we put it in there, and then we run the keys. We have all the keys, and then it's a process. Like we've been working with this for a while, right? So when we have the wood bolts, now we want to understand, is there any relationship between those traps and the wood? So what happened? I remember one grower asked me, Alejandro, you're promoting these traps? We have hundreds of beetles on those traps, but not all the 100 beetles actually attack my trees. Is there any relationship? What does it mean to have one beetle per trap? And I look at him kind of like, wow, that's one of the most profound questions that I have come from the growers. I love to talk to them. It's like, I didn't see that coming, but yeah, you got a point. So we run all these regressions. And then at the end, one adult per trap can go from 4 to 10 attacks. So one adult can be related. And of course, but correlation doesn't mean causation. But I understand that the big caveat of that. But one adult per trap can give you up to, if we use the lower end, will be 4. Again, what is the threshold in ornamentals? 0. So having one adult have the potential, if your trees are stressed, that you're going to end it with attacks. Now, for ambrosial beetles, the landscape ecology, it's way in advance in my lab, at least, in terms of trying to understand. We have already collected more data compared to the red-headed flea beetle, but see, you have a summary, a snapshot of the actual different spatial patterns in the field of ambrosial beetle. So the darker the color, the higher concentrations. And why do we want to understand that part? Is because we are trying really hard to use a trap and kill system. So using ethanol and vervenone, we really, really would like to come up with an attract and kill system. So spacing and the relationship with space is critical. That's the missing part that the entire group working on ambrosial beetle, we're trying to get to that point. So how to understand? We have a 2-pronged approach. One is the flying meal. That's the ambrosial beetle right there, going around and around and around in a controlled environment. That's a when you have the temperature and humidity, and it goes around and around. And actually, this one has been labeled when you walk through the hallway, the beetle torture chamber. Yeah, I'm gonna change that name. I'm not proud of that, but that's how it's been labeled every time I got visitors to our station. And I'm like, that's a really negative connotation. We're really learning a lot. So these ambrosial beetles actually can fly up to 500, on average, 460 meters times 3. So over 1,000 feet when you have a control. So that's the potential. Then we do the mark and recapture, very similar. But the beauty with this system is that we use ethanol. Instead of going randomly and using a vacuum, we got the ethanol trap. So whatever is marked, a proportion is going to go through those traps because they're, so our recovery rate, it's close to 15%, something like, so it's super higher compared to the other one. And the idea is the same. You shine a UV light, since it's a fluorescent. Our data show that up to 250 feet, we can recover ambrosial beetles that were marked. And we're going to increase that distance this year. Maybe 500 or 600 feet. It's difficult to try to fit those type of studies in wooded areas that we have around. And last but not least, the new invasive species in the United States that are actually affecting ornamentals is the box tree moth. On the left, that is a healthy boxwood, July 11. 14 days later, the boxwood plant was gone. The culprit, that beautiful. You see, I use those things, and the growers hate that because they look, an amazing caterpillar with the ability to defoliate, because there's some hundreds of those, and then that very conspicuous and big moth, the adult. One of the homeowners affected back in Niagara County in New York, this person was really excited, called me and said, Alejandro, I didn't know boxwoods promote the presence of beneficials. Can you elaborate? Yeah, I have this moth coming around, so I am so happy. I was like, can you show me what are you referring to? And we went to the boxwood. I shook that, and it was all box tree moth. It's like, well, sir, you know what? I have something to tell you. You better go to Home Depot and Lowe's and try to find something, because in 2 weeks, your box is going to be gone. What happened? So education was also part of the entire project that we did in New York, to present this information to the homeowners that were suffering from this. To the point that I tell my 2 kids, Joshua and Emma, that the true very hungry caterpillar exists. When we read that book, I said, the box tree moth now ate 2 oranges, right? And look at this. Caterpillars peeling the bark, going for the cambium, feeding through all that, and killing the entire shrub. So basically, you're going to lose, no matter if you leave it unattended, you're going to lose that. New Yorkers have this connection with boxwood because it's the only thing that's going to be green when they have a harsh winter, and they love it. The epicenter, ground zero for box tree moth in New York was this property in front of Lake Ontario. We went and visited. The owner, she used to have 352 boxwood plants. She lost every single one. She replanted every single one again. Now is spraying BT, now changing the cultivar. Now, like, she is an expert on box tree moth running. But actually, this is from her property. That shot, I remember vividly. This is what happened when you drive around that county. The boxwoods, the dead boxwoods, are ended at the end on the curbs. So 6 active quarantines in the United States. This is creeping up. We did a phenology study. We went every week. Why Virginia? Well, Virginia has one of the largest boxwood growers in the nation. And I was cornered to actually, hey, we're interested in knowing about this, so you better start learning about box tree. I was like, well, say no more. I should be able to. So we use the pheromone. As you see, we got a sticky cards to do a little bit of beneficials, and that's temperature and humidity data loader. Long story short, we were able to find 2 peaks of activity for adults at the top, And then the middle are the small from first to third to third instar, and then the one in the bottom is fourth, fifth instars. They will not, so the box remote will overwinter as third instar for the next generation. That will be able, so we were able to document 2 peaks of activity, meaning those 2 generations. We did a little bit more about what growing degree days, trying to know specific growing degree days based on previous publications. So we got the data with the temperatures, and we provide all this because this is a close collaboration with APHIS. So USDA, APHIS, and PPQ. Unfortunately, we didn't see any relationship when we trying to look between the beneficials we identify and those infested boxwood and the caterpillars themselves. Soon to be determined. They are what I call the new kid on the block. So basically, those enemies, they don't know their prey. So, all right, I'm gonna fly through this one because I'm a little behind. After knowing the past, I would like to use new tools and technologies. So here is an example to improve that management. Here's an example of using smart traps where we use different well, in this case, our photos, and the machine recognize each one of, because they have thousands and thousands of photos, and then recognize which ones are the pests, and they send you an alert to your phone to know how many insects you have in a day. That was a very great, we did the first trial when this company came to the United States in order to do this specific Lepidopteran back in Salinas. Then we got an array of beneficials that I'd like to remind the growers and anyone that I talk to that they're doing a great job. And that's what, so for example, here I have Kevin Hill, one of my collaborators, flying a drone with beneficials in it. So there is lace wings with rice hulls, HULL, the inner material. You mix that, you put it in the drone, and the drone basically disperse the beneficials. In the past, you're going to use people, a group of, labor. And then we humans, we try to cut corners. And it happens to me in Peru when I was have my crew of releasing beneficials. I'm going to see the holes in a pile at the end of the rows. And I was like, what's happening here? And nobody knew anything. The drone is not going to do that. The only thing the drone is going to do is like, oh, I need to change a battery. And then you bring it back home, change the battery, go back. You see, and they're dropping from the sky. Literally, that's why it's powerful. I think it's catchy. So what we did is I thought this was too good to be true. But at the end, we were able to show the growers that, yes, when you put some collection stations through the path of the drone, you're going to get what it's actually landing. And then you're going to get the lace wing eggs within those same samples that we were collecting. So at the end, we ran some sort of a demonstration project where we were able to show that either the lace wings that are deposited, or if you apply an organic certified pyrethrin, you will lower those populations of aphids per lettuce plant at the end. So it was very eye-opening. And then, the multiple growers, because that was only one, there were multiple, told me, Alejandro, do you think we're going to use something that doesn't work? We actually, we knew this. That's what I call this a demonstration project more than we would like to test. This one is mating disruption for box remote in Western New York. We use the dispensers of pheromone coupled with Bacillus thuringiensis applications. That's the dispenser right there. And then the idea is that you are gonna confuse the males because you inundate the system with pheromone. Here's some. We knock 220 doors to collect, actually enlist the homeowners to help us. There were 3 different parts of the town, different towns that we were able to put. So to give you an idea, this is one of the towns. The properties that are in green accepted. The properties that were in red, they say, no, I don't want you on my property. And then we put the dispensers depending on the area you have. Long story short, we're able to see, you see the flat line, that's for the treatment wear. And then the one that has the black dots is the one that are the controls outside the area. So we were able to successfully suppress not only the moth, but also, and I'm showing you the data, but damage was also decreased. And the number of caterpillars were also decreased as well when we put the combination of mating disruption, and bacillostringensing application. We did more, as usual. I am really interested in testing different chemicals and different settings to see which ones are the alternative. One of the things is application technology. We're trying to promote the idea to use drench or top dressing, which means that you have a granular product that you can put on the pot, and then having that instead of doing broadcast applications of this. Long story short, some of the products will work, some of the products will not work, and that's the intention of doing all this testing for telling the growers some of the suggestions. I promise that our group for ambrosia beetles are doing our best to try to find alternatives, because the only successful control tactic for ambrosia beetles right now is the spray pyrethroids. And we're trying different things. And the arrows are the pyrethroids, and the rest are, these are number of attacks. And we haven't been able to find one chemical that it could be a replacement for pyrethroids. Not yet. Stay tuned. We're not going to give up. All right. So as you see so far throughout the presentation, our target is the biology and ecology of pests. We are proposing, in the case my lab is proposing, to combine the pest control tactics, we're meaning validating existing ones or bringing new technologies. The research is under field conditions, and what we go is across systems or across stakeholders and across the pest issues, where we do the biology and monitoring, the cultural and biological control, and also the chemical control, and looking into, I didn't present any data, but we're looking into some insecticide resistance, especially for red-headed flea beetle. The need assessments for growers is actually a great guide for, in the case for my lab, where we rank and we have those priorities where we kind of put the effort to conduct the research. And ultimately, we would like to provide those science-based solutions as a technical support. So to wrap it up, in summary, what I present today, red-headed flea beetle, Japanese maple scale, and ambrosia beetle are key pests in Virginia. The box tree mold is a new invasive that it's inching this way in, and we actually were able to team up with USDA, APHIS, and PPQ, and now we have a compliance agreement that actually avoid the limiting of the commerce of boxwood in the United States. Unlike Canada, when they have that problem, they stop selling boxwoods, and that hurt a lot. And here in the United States, we have a compliance agreement, so growers have to go through that certification and specifically comply, and they will clean the product and they can sell it. So that was huge. Biological control is also one of the priorities within my lab, especially augmentative biological control. I do have a drone that releases beneficial in my lab. Is one version, and I'm trying to expand the reach of that. So I got some crazy collaborators that are believers of these things, and we'll try new things. So stay tuned. We also, in my lab, we bring in these new technologies. There's remote monitoring, there is mating disruption, and other things to expand that toolbox for the growers. And definitely IPM, it's what we would like to see to reduce both risks, the economical, but also the environmental. Recognizing the chemical control is the most used tactic, and since we have to be aware of that, we really need that expansion of the toolbox. With that, that's my house. No, it's not. This is actually from but I want to finish. This is what it brings to the green industry. I want to be barefooted there playing with my kids. This is what we mean to society, to the community. This is the joy of you doing gardening with your own hands. This is what the entire group would like to keep that. And well, pest gets in the way and reduces profits. That's the idea, to provide those science-based solutions. With that, thank you.